Hay que tener en cuenta que tanto en la FDA como en muchos organismos, quien manda es el jefe. Y si bien, digamos, estas organizaciones tienen científicos y gente que valora la información que ofrecen las corporaciones, todas esas, digamos, observaciones y advertencias que hicieron ver científicos conscientes sobre este tema dentro de la misma organización fueron obviadas, porque como les digo, ahí la decisión la toma el jefe, y eso es lo que ha venido pasando, como lo vamos a ver, nos lo va a explicar el doctor Druker en su presentación. Sobre este tema, que es uno de los que la gente, digamos, más pregunta y se interesa, ¿verdad?, de eventuales riesgos e impactos a la salud, les quiero avisar que este viernes a las 6 de la tarde en el auditorio 180, la máster Mildred García va a dar una charla sobre un experimento, digamos, el experimento más completo que se ha hecho con animales en experimentación, que duró 2 años. La mayoría de los permisos para desregular un alimento, un cultivo transgénico, en toxicología hasta la fecha han sido de 3 meses, y este es el primer estudio que, digamos, se sale de ese protocolo, llega a 2 años, y donde se demuestra que casualmente, ¿verdad?, es a partir de los 3 meses que empiezan a verse los problemas de salud en estas ratas con las que se experimentó con una variedad de maíz transgénico. Entonces están cordialmente invitados, digamos, para quienes quieran conocer un poco más en profundidad sobre esta investigación de un grupo de investigadores franceses liderados por Seralini, que ha sido muy controvertido por quienes no les interesa que ese tipo de información se dé, ¿verdad? Quien ha hecho posible, digamos, la presencia del doctor Druker en nuestro país, pues ha sido la Fundación Pax Natura, y aquí tenemos con nosotros al señor Randall Toppenroth, que es su presidente en la actualidad, y realmente pues para nosotros es un honor que hayan escogido nuestro país como el segundo país en el mundo para presentar esta publicación. Que como me estaban diciendo ahora, más o menos dentro de un mes vamos a tener disponibles copias en inglés, y se espera que en unos 6, 7 meses ya esté disponible la versión en español de este libro. Entonces, para que estén atentos para cuando llegue este libro. Ahora lo voy a volver a presentar. Bueno, ya está aquí. Ya tenemos al doctor Druker con nosotros. Antes de darle la palabra, bueno, presentarlo. Él es un abogado estadounidense de interés público, filósofo de la Universidad de California en Berkeley. Posteriormente hizo sus estudios de doctorado en Derecho en esta misma universidad. Es miembro de la revista jurídica California Law Review, así como de la Orden de COI, que es una sociedad jurídica de honor. Y es el cofundador y fundador principal y director ejecutivo en la actualidad de la Alianza para la Biointegridad. Antes de darle la palabra al doctor Druker, quisiera invitar al doctor Luis Bernardo Villalobos para que dijera algunas palabras y agradecerle a él y a su equipo aquí en esta sala por haber tenido la gentileza de habernos facilitado este lindo auditorio.
Muchas gracias al doctor García. Quisiera dar la bienvenida al doctor Druker y decirle que es un honor para la Universidad de Costa Rica y particularmente para la Facultad de Medicina. Quería decirle al doctor Druker que es un honor para la Universidad de Costa Rica y para la Facultad de Medicina contar con sus reflexiones acerca de este tema tan importante que tiene que ver justamente con la biointegridad. Quiero agradecer a la organización Pax Natura también, y al doctor García, y a todos los grupos que han difundido esta conferencia, y a todas y todos ustedes por estar acá. Yo creo que el tema que va a tratar el doctor Druker es fundamental, es un tema muy importante para la salud pública del mundo, y igualmente es un tema en el cual nosotros desde la cátedra Dr. Rodrigo Loría Cortés de la Facultad de Medicina estamos comprometidos a difundir. La cátedra en la cual se enmarca esta conferencia es una cátedra creada por la Facultad de Medicina para exponer temas de interés público en el campo de la salud y el bienestar comunitario. Es una cátedra que aborda temas que tienen que ver con la interrelación ambiente y salud, con la bioética, con la trata de personas, con toda una serie de problemas que aquejan a la sociedad contemporánea. Y este es un tema fundamental que queremos que quede expuesto, y también queríamos comentarles que estamos transmitiendo por streaming en tiempo real, así es que posiblemente algunas personas fuera del país o dentro del país también están teniendo la oportunidad de escucharnos y vernos. A estas personas les decimos que pueden dejar sus preguntas en el chat que ahí aparece en la página de la Facultad de Medicina, y posteriormente se las haremos llegar al doctor García para que se las haga llegar al doctor Druker. Esas personas pueden tener la plena seguridad de que sus ideas también podrán ser transmitidas al doctor Druker. Muchísimas gracias, doctor Druker.
Bien, entonces la dinámica va a ser, el doctor Druker va a hacer una exposición y después de esta vamos a tener un conversatorio, más o menos como hasta las 11:30, porque tenemos que desalojar la sala porque hay otra presentación después. Entonces, esa va a ser la dinámica. Doctor Druker, le escuchamos.
Thank you very much. Can everybody hear me all right? Okay, thank you. Well, I'm very grateful for the opportunity to speak in this very fine facility in this beautiful country and to such a good audience. And my plan is to speak for perhaps 35 or 40 minutes and then take your questions. And I assume there will be many questions, and I think we can have a lively question and answer session. Now, let's suppose that a radical technology gets developed through which novel forms of food can be produced. And suppose that although this method is highly unnatural, its proponents are somehow able to convince most people that it's essentially the same as producing new varieties of crops through conventional breeding. And let's further make the supposition that the very first ingestible product that this technology puts out, let's say it's a food supplement, let's say that this first product causes a major epidemic that kills dozens of people, seriously sickens thousands more, and that many of these people who don't die but are seriously sickened remain physically impaired for the rest of their lives, hundreds of them. And let's postulate an additional fact. Let's suppose that researchers discover that the product contains a highly unusual mix of contaminants, at least one of which is deadly at extremely low concentrations, so low that most chemicals present at such a level would be harmless. And let's further imagine that the evidence as a whole strongly points to the new technology as the cause of that lethal contamination. Moreover, let's suppose that the first whole food produced by this technology causes a troubling pattern of stomach lesions in the laboratory rats that have to eat it, and that in another group of rats eating this product, the food produced by the new technology, 7 deaths occur in that experimental group within the first 2 weeks, while only 1 death occurs in each of the 3 comparative control groups that were spared consuming that novel product. And to further foul the picture, let's assume that government scientists review the test results and conclude that they raise a safety issue that has not been satisfactorily resolved. And to make things even more dramatic, let's suppose that another division of government experts who review a separate aspect of this product declare that that feature in itself poses a serious health hazard. Okay, now that we're going and warmed up, let's assume that a task force of government scientists thoroughly assesses this technology and concludes that it significantly differs from conventional modes of food production, that its products entail higher risks, and that each therefore needs to be subjected to rigorous safety testing before it is allowed on the market. Furthermore, why don't we make things even more ominous by imagining that the first food produced by this technology that is subjected to truly rigorous testing by independent experts who are free of industry influence yields very troubling results, and that because the study is of high quality, it gets published in a prestigious scientific journal. And now that we're becoming bolder, let's suppose that many other well-conducted studies on the foods created by this technology detect significant adverse effects on the lab animals, and that one long-term study finds serious injury to the livers and kidneys, abnormal onset of large tumors, and increased onset of mortality. Let's further envision that when independent scientists scrutinize the research that has been performed by the corporation producing the foods, they in several cases find that the data that was declared to support findings of safety actually raise serious doubts. And to make the plot even more sinister, let's suppose that in virtually every case of adverse results, there is good reason to think that the results have been the result of some aspect of the technology itself, and that similar deleterious disruptions could have occurred in other foods produced by it. So if such a technology actually existed, if such a technology actually existed and the suppositions that we've been going through actually had occurred, how do you think society should deal with it? I assume that just about everybody here would agree that the foods it creates should be banned, and that the enterprise that creates them should be stopped. And I doubt that anyone would consider that to be an extreme or unreasonable course of action. In fact, I think most people would suppose that in such circumstances, that particular policy would be fully justified and it should be decisively enacted very quickly. And just to be clear, that policy that would seem just and appropriate and necessary in almost everyone's eyes would not be to stick a label on those products, but to ban them completely. Well, as surprising as it may seem, especially to people who have been trusting what the United States government has been telling them, or what the scientific establishment has been telling them, such a technology does in fact exist. And every one of the suppositions I've just set forth is not a mere supposition, but a rock-solid reality. And that technology, of course, is recombinant DNA technology, which is commonly referred to as genetic engineering. And all of those supposedly hypothetical situations have actually happened. Every one of them has actually happened. And they're concrete events that are unshakably linked to the genetic engineering of our food. Now, most of you probably were not aware that all of those suppositions, which are really realities that I just went through, actually did happen and are linked to genetically engineered foods. And that's because there has been a massive campaign of disinformation and suppression of information so that you won't know that. And of course, the people in this room, many of whom didn't know that, are among the most well-educated people about this topic. You know, you either are students at or teach at a major university. I'm sure many of you have been studying this issue. I know you're very concerned about it, but I'm almost sure that you were not aware that every one of these situations I just described actually has occurred, but it has. My book documents it very thoroughly, and I'd like to now go back and just go through all of those and explain briefly what has happened and how they have been linked to genetically engineered food. Well, the first one we know did happen. The proponents of genetically engineered food have been able to convince the vast majority of people in governments around the world, your government, many people in your government, governments around the world, scientific universities around the world, the media, the public in general, that it's nothing really that unusual, that it's essentially the same as traditional breeding. And in fact, a statement that one hears often from proponents is it's just a minor extension of traditional breeding techniques. That, to say that it's a minor extension of traditional breeding techniques and essentially the kind of genetic modification that's been occurring naturally for centuries and centuries, that is such a gross misrepresentation of reality that when it comes from people who are agronomists or biologists, there's good reason to think that a lot of people are lying. Because when one understands the facts about how radical a departure this technology is from what occurs within pollen-based breeding, sexual reproduction, it's ludicrous. It's ludicrous. In fact, one of the many scientists who never got on the bandwagon to promote these foods and to try to put out that kind of misinformation, George Wald, who for many years was a professor of biology at Harvard University. He won a Nobel Prize. He's no longer with us, but he was alive during the initial phase of the genetic engineering revolution, and he termed it the biggest break that has occurred in nature in human history, the biggest break in nature. And then in contrast to that, you have people saying simple extension, part of an unbroken continuum. No, no, it was not only a break in the continuum, but according to this Nobel laureate, the biggest break that's ever occurred. And he went on to state it should not be confused in any way with any interventions mankind has previously made in the natural order. This is by far the most radical and the biggest break. And there are many other scientists who have said the same thing. And there's, well, my book goes into great detail explaining why this technique is so different, so radically different, and why it entails unique risks. Now, that second point that I brought out as a supposition initially, that the first ingestible, the first edible product of this technology causes a major epidemic, kills dozens of people, maims thousands. How many of you actually knew that the very first edible product of recombinant DNA technology did cause a major epidemic that killed dozens of people and wrecked the lives of hundreds of people? How many knew that? Almost no one. In fact, I didn't see a single hand go up. If none of you knew it, think of the situation of the average journalist and average person, because again, as I mentioned, you're the elite. You're university students and professors. You're concerned about this issue. You have tried to become informed. That shows you how efficient, how effective, and I would say diabolically effective that disinformation campaign has been. If you didn't know it, because it's the truth, there's a whole chapter in my book devoted to it. Back in 1989, a supplement of the essential amino acid L-tryptophan hit the market in the United States. Now, what was unique about that was not that an L-tryptophan supplement was being marketed. L-tryptophan supplements had been marketed very safely for many years in the United States, throughout the world. As I said, it's an essential amino acid. It's in a lot of our food. And in its natural form, it's very healthy. And even in its supplement form, it was very healthy. There had been no linkage of any L-tryptophan supplement with any ailment up until 1989. But in 1989, one form of L-tryptophan supplement caused a major disease. More than that, a major epidemic of a novel disease, a highly unusual disease. It's given the initials EMS. I don't pronounce it very well, so I'm not going to try. If anybody can pronounce it, you're welcome to, but it wouldn't really matter. It's a unique disease, and most of you are unaware of it probably anyway. And of course, unaware that it was linked to this particular supplement, because what was unique about that particular supplement of L-tryptophan is that it was produced through bacteria that had been genetically engineered. That's what was unique. All the major manufacturers of L-tryptophan supplements were in Japan, and they were using bacteria that naturally produce L-tryptophan, and they were producing the L-tryptophan through the bacteria, then taking the broth, putting it, the fermentation broth, they were using fermentation, then putting it through filtration, and then once it got to the level of standard pharmacopoeia purity, then they would market it, and there was no problem. Now, as many of you who are pharmacists know or have a background in that, it's just about impossible to get any product completely pure of impurities. That's why you go for the pharmacopoeia standard, which is, I think, over 99% pure, somewhere in there. But any impurities left are usually at such a low level anyway that even if they're somewhat harmful, they're at too low a concentration to create harm. this particular supplement produced from genetically engineered bacteria was very different. By the way, the manufacturer used genetic engineering in the bacteria to dramatically increase the rate at which L-tryptophan would be produced. Basically, those bacteria, and by the way, those bacteria were not transgenic in the sense that the key genes were copies, just extra copies of its own genes. So it's important to realize just transgenics is not the main problem. It's the recombinant DNA technology itself. It can disrupt the genome of the target organism in several ways and disrupt the metabolism of that organism, which apparently it did in this case. I'll discuss that just a bit more and then move on. So these bacteria were being given extra copies of the genes that are involved in L-tryptophan synthesis. And then, actually, this had been going on for several years. And it turns out that this disease had actually been created earlier, induced earlier, but at such a low level it didn't show up. It wasn't until the epidemic that scientists were able to finally understand something weird was happening, and they were able to link it to that supplement. But it took quite a while for them to do that, even with a novel disease. But the fact is that not only were extra copies of the genes given, each generation, each new version of the supplement, they used a more powerful form of genetic engineering to increase the rate of L-tryptophan production, because the company was focused on economics, not safety. And they thought what they were doing was going to be safe. You know, they were just giving the bacteria extra genes of their own genes. But the last form of the supplement that caused the major epidemic, to increase the rate of L-tryptophan production even more, a promoter from a virus was put in front of the set of those genes and basically that promoter pumped up the production round the clock, because viral promoters keep expressing their gene, because that's what a virus does. It's acting completely outside the regulatory system of the target organism, and that's what was happening. So this final, the version of the L-tryptophan supplement that caused the major epidemic, that raised the level of incidence of the disease to a high enough level where it was finally picked up by epidemiologists, was transgenic in the sense that a promoter, and maybe not all of you know genetics, but in any event, the promoter is the bit of genetic material, DNA material, that enables a gene puts the gene into an expressive mode so that RNA can actually be transcribed from that gene. And by taking a promoter, removing the native promoter from the from the bacteria, and putting a promoter from a virus there, what it meant was not only was the production of L-tryptophan being increased beyond the normal, but the bacteria were being forced, and that's not, forced is the correct term, because that's what the viral promoter does. It commandeers the gene expression machinery and forces it to behave the way the viral promoter wants it to. In a sense, the viral promoter is saying, basically, keep expressing this gene morning and night, non-stop. That's a highly, highly unusual situation. So what you have, and it appears from data within the company's own files, that there was stress being put on those bacteria. Quite a bit of stress, it appears. My book goes into it. I won't have time. I could spend the whole time speaking to you on this, but I need to move on. But it's important to know that that product tested pure. It passed the pharmacological testing for purity. And yet there were contaminants within it that were so deadly at such a low level that they created a major epidemic. And in fact, very unique, and to the evidence as a whole. And by the way, another unique feature of my book is I called up the researchers at the Mayo Clinic in Minnesota in the United States. Many of you might have heard of the Mayo Clinic. It's one of the most prestigious medical clinics and research organizations. And I knew that there was a team of researchers there that have been researching